So we're going to start Unit 6, Lecture 3, and for this lecture we're going to focus on the nutrients that are involved with the blood. And primarily we're going to be discussing vitamin K and iron. Those are usually the two that you think of the most with the blood. And we're going to touch on copper, because copper, one of its main functions is it assists iron, and without copper you're not going to have iron incorporation into heme. There are a few other vitamins that if you are lacking, you will get symptoms involving anemia, but we're going to be discussing those later on in the semester. So as I mentioned, we have numerous nutrients that are required for regulating blood composition and fluid balance. We have protein that's needed, that's going to be making serum amyloid or serum albumin, and various acute phase proteins such as serum amyloid A and C-reactive protein. And then our electrolytes, our sodium, potassium, and chloride are helping maintain osmotic pressure and keeping our acid-base balance. Iron is probably the nutrient that most people associate with the blood because that's what's our hemoglobin is utilizing to carry our oxygen around. We have copper, folic acid, B12, and vitamin K. So with this lecture, we will be discussing iron and vitamin K. So before we can really talk about the nutrients, we need to talk about what blood is. So what are some of the blood proteins that we're referring to? So we have three main groups. The first group is albumin, and albumin is what the protein that makes up about 60% of the plasma protein. It is responsible for keeping the osmotic pressure. So if you have inadequate albumin, that's when you're going to get diseases or disease states such as edema and ascites. So many times, basically, if you go back through the notes, times in which I say edema or ascites is a deficiency, it can come back to how is it affecting your blood albumin or your osmotic pressure of blood. Albumin is also important for transporting copper, so this is how our copper is transported in our blood, and it also transports our fatty acids. So our fatty acids don't solely go through our lipoproteins. They can be conjugated to albumin and be transported that way. Our next classification is our fibrinogen proteins, and these are the ones that are involved in blood clotting. So you cut yourself and you start to bleed. What's stopping your blood what's stopping you from bleeding to death is due to the fibrinogen that is formed, our fibrin, and that blocks the hole and allows the body to heal so you don't bleed to death from a paper cut, for instance. And then the third category is our globulins, and this is mainly going to be our hemoglobin for blood. All these proteins, most of them are synthesized by the liver. Most are conjugated to glycoprotein or to carbohydrates, making them glycoproteins. The only exception to this is our albumin. Albumin is not a glycoprotein. It's not conjugated to a carbohydrate, but the rest of them are. So we're going to focus first on vitamin K. And vitamin K is most notably involved in blood clotting. There are other functions for vitamin K, but this is the one that everybody knows pretty well. Or has heard of, is vitamin K's involvement in blood clotting. We have two major forms of vitamin K. We have naturally occurring forms, which include phylloquinone, which is also known as K1, and that comes from plant origins. And within this naturally occurring, we also have menaquinone, which is a bacterial origin, also referred to as K2. And then we do have synthetic menadione, which can, sometimes you may see it referred to as K3. So this is typically, menadione is what you're going to find in your multivitamins the majority of the time. So here you can see the differences between the phylloquinone and the menaquinone compared to menadione. And not important for this class to know, but there are two different types of menaquinone. So you have menaquinone 4 and menaquinone 7. And the main difference between phylloquinone, the plant version, and the menaquinone is you could see especially with menaquinone 7, it has significantly more double bonds on the structure compared to the phylloquinone. Now the menadione does not have this carbon chain that you see in phylloquinone and menaquinone. So basically, when we absorb the menadione and it travels to our liver, we actually add that carbon chain onto the menadione. So our vitamin K is a fat-soluble vitamin, so therefore it requires the presence of fat and bile salts for optimal absorption. If you don't have fat and bile salts, it will not be absorbed very well. And basically, and this varies somewhat depending on the species, but for the most part, all throughout the GI tract, we can absorb vitamin K. So it can be absorbed all throughout the small intestine, and you can also absorb it from the colon. So some texts say that it's the proximal intestine, indicating mainly the jejunum, and you'll have the majority of it absorbed in the jejunum, but we can't disregard the bacterial menaquinone that's able to be absorbed from the colon. And that's with all species. So, and because we're able to absorb it from the colon, we actually can meet the majority of our dietary requirement of it from our microflora synthesis of the menaquinone without having to practice coprophagy, which is different compared to some of the other microflora synthesized vitamins. When we are absorbing it, we utilize active transport. So both the phylloquinone and the menaquinone utilize active transport, and it has to have a carrier protein for it to be absorbed. It really has not been elucidated very much, the actual carrier protein, because vitamin K can be absorbed all throughout the GI tract, and we have these various forms. It makes it a little more difficult to study. The majority of what we do know comes from studying phylloquinone. It's very difficult to study the menaquinone absorption. The menadione, the synthetic, can be passively diffused. So our K3, because it does not have that large carbon chain, can just passively quickly diffuse through the cell membrane. And once we get into the enterocyte, we have it that the vitamin K can be transported by the lipoprotein, so it enters the chylomicron along with our other fat-soluble vitamins and our triglycerides. And as our chylomicron goes around, when it gets back to the liver, our vitamin K can be stored within the liver also. And when we have adequate amounts of vitamin K, we are able to get rid of it. So when we are metabolizing it. We have about 20% of it ends up being broken down and can be excreted in the urine. And then the other 80%, we actually excrete it with our bile salts. So we break down the metabolites and they leave with the bile salts and are excreted within the feces. So here on the bottom, we have a diagram showing our dietary vitamin K coming in. It enters with mixed micelles here, and then that can enter into the enterocyte. We have, this is our chylomicron that's formed here. That's able to enter the lacteals, goes into the thoracic duct, goes into the blood, carried all through the tissues, and that's how we're transporting our vitamin K around. So the main way in which vitamin K functions is that it actually is a co-substrate in the post-translational oxidative carboxylation of proteins that have select glutamic acid residues. So it utilizes many of these reactions, end up taking the glutamine on the protein. So for instance, here, this example is using prothrombin. So we have glutamine on the protein, and we switch it over to glutamic acid. So we're going from an H here to an acid. And it does that on select glutamines within the protein. So it utilizes the vitamin K dependent carboxylase, converting glutamic acid to gamma carboxyglutamic acid. And so there are various proteins that utilize this, and the main ones that we always think of are the coagulation ones. So there are 4, so there are actually 13 proteins that are involved in coagulation. So we have 4 of the 13 need vitamin K to convert the glutamine to glutamic acid. So that is factor 2, which is also known as prothrombin, factor 7, factor 9, and factor 10. So all of these are involved in coagulation. And we also have some others, so proteins C, S, and Z, and these are involved in anticoagulation. And then finally, we have osteocalcin. So this is one of the overlooked functions of vitamin K that is actually pretty critical for proper bone function and mineral density. You have to have vitamin K to activate osteocalcin. So here, just showing this reaction, we have our, and this is called the epoxide cycle. So our vitamin K is our vitamin K hydroquinone is converted to vitamin K2,3 epoxide. And this actually also requires niacin in the form of NAD and riboflavin as FAD to help this reaction occur. And then we have our vitamin K is regenerated using vitamin K epoxide reductase and vitamin K reductase. And we'll talk about those more in a few minutes. So before we get into the clotting and how the clotting factors all work, I'm going to spend just a minute or two talking about the vitamin K's involvement in bone mineralization. And we may touch on this again when we're talking about the skeletal integrity. So here we have our pre-pro osteocalcin. So this is made by the osteoblasts. And the expression of this is actually controlled by vitamin D. So vitamin D is controlling this. So this is one of the ways that vitamin D is controlling bone density also. So we have our signal peptidase that's going to end up converting pro osteocalcin to pro osteocalcin. And then we have our vitamin K carboxylation that's occurring, which is now activating our osteocalcin. And then our active osteocalcin is able to bind calcium. So this is it has a very strong affinity for calcium, and that allows for bone mineralization. So you have to have active osteocalcin before you have mineralization. So if you end up blocking vitamin K function, for instance, with the drug warfarin or Coumadin, you're actually going to inhibit this, and you're going to have poor bone mineral density. So here we have a summary of the blood clotting cascade, and there are several phases involved in blood clotting, and it can be activated. We have both an intrinsic system and an extrinsic system. So our intrinsic system is going to be more of if you get a bruise, how your body is controlling that, versus extrinsic system where you trip and fall, skin your knee. So that would be extrinsic. So internal bleeding versus external bleeding is basically how you can separate it out. So we have several phases. The first of them is the formation of thromboplastin. So this is this component right up here. So we've and this is a very in-depth cascade that we are not going to discuss. That's what your physiology classes are for. But here you can see our vitamin K is required to change that glutamine to glutamic acid and on the plasma thromboplastin component with number 9. And then this actually ends up activating a bunch, and eventually we get because this really likes, our vitamin K really likes calcium, so we need calcium and phospholipid, and we end up activating our thromboplastin. So first we form it, then we activate it, and then we have our second is our factor X. So we are activating it through our factor X. So this is number 2. And then we have the formation of the thrombin. So we add the proacelerin and a phospholipid, Defilin. You don't have to know the particular type of phospholipid, just know that we need active preacelerin and a phospholipid to where we actually form thrombin. So this part is referred to as prothrombin. So we're forming that here, and then we have the formation of our thrombin. And then we need our thrombin to where we can activate our fibrinogen. So our fibrinogen and our thrombin come together, and we get the formation of fibrin. And this is actually what is basically what's covering the hole, and it's very sticky. You can almost think of it as a spider web type contraption that's going to be making everything stick together, and that's what's blocking the hole. So here we have a magnified blood clot, so you could see all of our red blood cells and all this web component, all this white part. This is the fibrin. So you can see within this fibrin structure that forms, we have red blood cells, we'll have white blood cells, platelets, etc. And they all get stuck in there, and that's what's forming your lovely little scab that you enjoy picking so much. So when we have deficiencies in vitamin K, first, primary deficiencies are extremely rare because we are able to absorb vitamin K from the microflora in the colon. Humans can meet all their vitamin K requirements basically based off of what the microbes in your GI tract are synthesizing. In addition, it's found in all your green leafy vegetables, so all of those have adequate amounts of vitamin K. So as long as you are consuming a well-balanced diet and you like your green leafy vegetables, you should be adequate with your vitamin K. Now, the majority of the times when we see deficiencies, it actually presents itself when we have some sort of fat malabsorption that's occurring. You can also encounter vitamin K deficiency if you are utilizing chronic antibiotic therapy. So because vitamin K can be stored, it takes a few weeks of utilizing antibiotics before you would see a deficiency. And then you can encounter it if you are utilizing an anticoagulation therapy, including warfarin and Coumadin. So here we have a few pictures on the lower right-hand side. You can see this young boy. He has a runny nose, so that can indicate some sort of vitamin K deficiency. You bruise very easily, so that's our internal hemorrhage. You have hematomas, bruises, etc. So we also have prolonged clotting time. So if you skin your knee where it normally may take a minute and 30 seconds to clot, it may take 3 or 4 minutes to clot. So those are going to be some of the main signs. And then if it goes on for a prolonged period of time, you're going to see a decrease in your bone density, and that's due to the way in which vitamin K is involved with the osteocalcin. So we do encounter vitamin K deficiency in ruminants. Actually, this is how we discovered warfarin or Coumadin. So in the early 1900s and around 1918, I believe, there was many farmers that were planting sweet clover in the pastures for their cattle, and they had a plague of these cattle were just dying. They were hemorrhaging, and so many of these farmers were very upset, and they couldn't figure out why. And it actually took about 15 years for scientists to catch up and answer this problem. So it eventually ended up being to where there was a farmer that had sweet clover, and they baled it, and this sweet clover got moldy. And they also brought their dead cow to the University of Wisconsin in Madison, and it was there that they were actually able to isolate the compound coumarin. So basically, the mold within the sweet clover produced the compound coumarin, and the mold does that because it's actually an antifungus. And that's how it was discovered. So upon further review, it was discovered that this coumarin actually blocks the epoxide reductase and the quinone reductase. So here we have that epoxide cycle again. So we have our vitamin K hydroquinone going around to our vitamin K epoxide. And when we go to have it go back to vitamin K, we can see that our warfarin here is blocking our vitamin K epoxide reductase and our quinone reductase. So basically, we're not able to regenerate our vitamin K, and therefore we end up just excreting or eliminating our vitamin K epoxide. And if we keep on doing this and are unable to recycle it, then we're going to end up in a deficient state. So that is how we discovered Coumadin. And interestingly, it wasn't, it quickly was developed and we found the best form, and it was used for rat poison. So if you ever use Decon or whatnot, you are using warfarin or Coumadin now. And so it was only used as a rodent killer. And it wasn't until the 1950s to where it started being evaluated as a drug, the warfarin, to thin individuals' blood. And it was thought that it would be too toxic and would kill people. And I believe it was in like 1953 that there was some individual that actually tried to kill himself with warfarin and failed. And that's when the federal government decided to start utilizing it for and evaluating it for as a blood thinner for human patients. And then President Eisenhower started in 1955. He started taking Coumadin after he had a heart attack. So here you can see a couple pictures of some poultry that are deficient in vitamin K. You can see the breast that's all this blood that was hemorrhaged out. So before it was necropsied, it was all bruised. And here we have this chicken that you can see that the wattle here is very pale. It's not just the poor quality of the picture, but this wattle is pretty pale. So that's an indication that the chicken may have vitamin K deficiency. So if you just see one, there could just be sick, where if you see the whole flock or a significant amount of the flock with pale combs, then it very well could be a vitamin K deficiency. Toxicity in vitamin K is typically pretty rare, especially if you're utilizing the natural sources. The phylloquinone and the menaquinone are basically non-toxic. You're not gonna ever have any problems. The place that you run into problems is with the synthetic. The menadione is highly toxic, and if you end up taking too much, some of the symptoms include anemia. You can have acute renal failure. You'll find stiffness, colic, and laminitis within horses. So that's basically all on vitamin K. Now we're going to switch gears a little bit and talk about iron, which is the nutrient that most people associate with blood. So iron is present in a variety of inorganic salts, and one of the reasons is because it has so many different valencies. So the main form that we're going to be focusing on is the trivalent form, or ferric, which is Fe3+. And then we have our reduced form, which is ferrous, Fe2+. And these are the only ones that are absorbed from the intestine. It makes up the essential component to carry oxygen within the body. In addition, and this is what the majority of us are thinking about when we think about iron, is how it assists hemoglobin with carrying oxygen. And then, but there are other functions which include it's a cofactor for enzymes involved in many of the redox reactions, specifically many of the metalloproteins. So iron is critical for adequate oxygen transport, and we have various iron-containing proteins that help carry and release oxygen all throughout the body. So the one that we most commonly think of is hemoglobin, and hemoglobin makes up 95% of the protein within the red blood cells. So this RBC is a red blood cell, and consequently it is making up about 60%, or holds about 60% of the body's iron. Now, the second most prominent protein is myoglobin. So myoglobin is found within our muscle cells, and myoglobin is making up 1% of muscle protein and holds 4% of the body's iron. And next we have our ferritin. So ferritin is found within the liver, the bone marrow, and blood, and the intestines, and it has a range between 5 and 30% of the body's iron. And then finally, transferrin, it transports the protein in the blood, it is the transport protein in the blood and extracellular fluids, and it contains 0.1% of the body's iron. So here we have a diagram of hemoglobin. So you can see our hemoglobin has 4 subunits that make up the molecule. So we have 2 alpha chains and 2 beta chains, and each chain has an iron in it. So here we have 1, 2, 3, 4. So 4 irons per hemoglobin, because hemoglobin is made up of 4 different proteins. So how is iron absorbed? Well, iron is absorbed, and it mainly occurs within the duodenum and the jejunum. And as I mentioned before, we have the different types of iron. So we have ferric and ferrous. Now, one of the main things that's going to affect the bioavailability of iron within your food is determining which type of iron you have. So all ferric iron, this Fe3+, needs to be reduced to ferrous. So the oxidized ferric needs to be reduced to ferrous, and only ferrous is able to be absorbed. And you'll notice here in this picture that we have iron from food. Any of our iron that is incorporated into heme or myoglobin is going to be more readily available, so it can come through this heme transporter. And this is one of the reasons why iron is more available when you obtain it through animal products as opposed to plant products. Plant products are going to have a lot more ferric iron as opposed to ferrous iron. So then we have to reduce it down to ferrous, and then we have to absorb it. So heme iron, really easy, comes through the heme transporter, enters into the enterocyte. Our non-heme iron, especially if it's ferric, we utilize iron reductase, also known as duodenal cytochrome B. So ferric reductase is what is reducing ferric iron to ferrous iron, and then we are able to absorb the ferrous iron within the intestine. Once it's inside the enterocyte, our iron has to be bound. As I mentioned before, iron is going to be one of the main minerals that is going to be causing oxidation and cause peroxidation within the cells. So we have to bind it. So pretty rapidly, as soon as it enters into the enterocyte, we have it bind ferritin. So this is ferritin found within the enterocyte. And then it is oxidized back to ferrous, or back to ferric iron, and it leaves. So we have our ferrous iron leaves, and it's converted back to ferric, and it binds transferrin. So transferrin is what is transporting our iron throughout our tissues. And our iron is stored within the liver, and it's stored in the hepatocyte. So this is our liver cell, and it's stored with ferritin. So transferrin transfers the iron to ferritin, and is stored in the hepatocyte. I'd like to draw your attention here to this hepatocyte. You could see when we have adequate iron within our liver, our hepatocytes start secreting the protein hepcidin. So this is actually going to inhibit and cause a downregulation of the ferroprotein 1, which is the transporter that's going to cause the transportation of the iron out of the enterocyte. So if we have hepcidin, it's going to decrease transport of ferritin, it's going to decrease the transport of the iron from the enterocyte. So that's how we're controlling and trying to limit the amount of or excess iron that we have. And we have other things that can also affect the absorption of iron. So we have phytates, as we mentioned before, tannins, which are polyphenols found within plants, and acids are able to inhibit iron absorption. So here's some factors that influence iron absorption. We have, as I just mentioned, these inhibitors, the phytates, tannins, clay. If you consume too much iron, then you're going to have a lot more of the hepcidin that is secreted. So too much iron is going to inhibit more iron absorption. We also have here the physical state, the bioavailability. So our most readily available is going to be heme. Then we have the ferrous, and then ferric iron is the worst. So our ferric iron is the least available to us, and we have to reduce that back to ferrous. We have competitors. So these are, if you look on the periodic table, they have similar charges within them. So our lead, one of the reasons why lead is so poisonous is it can replace iron. It uses the same transporters, and it can bind ferritin and transferrin. So that's why lead is so poisonous. We also have cobalt and manganese and zinc can also inhibit iron absorption. And then our facilitators include ascorbic acid or ascorbate, which we'd mentioned before, citrate, amino acids, and if you have an iron deficiency. So if you are deficient in iron, you're going to have a low level of the hepcidin, so that's going to cause an increase in the upregulation of your transporters. So then you'll be more able to absorb iron. It'll do it more efficiently. Now, the ascorbic acid and citrate, these are mainly increasing the absorption by reducing the ferric iron to ferrous iron. So then it allows for the better absorption. Other inhibitors of iron include some organic acids. So our ascorbic acid and our citric acid are going to increase absorption. There are some other organic acids that will actually inhibit the absorption. You have phosphates that can inhibit absorption. As I mentioned, the phytates, oxalates, gossypol, tannic acid, and aflatoxins. And aflatoxins are chemicals that a lot of molds produce, and there are hundreds of different aflatoxins. So it all depends which particular aflatoxin that mold is making, and some of those can inhibit the absorption of iron. So here's a picture of what phytate looks like. As throughout the rest of the semester, I'm probably going to be discussing phytate a lot because it does inhibit the absorption of many of our minerals. So here you can see the inositol ring. That's the main component. On each ring, we have a phosphate molecule, and these phosphate molecules are able to bind the positively charged ions. So we have zinc, calcium, magnesium, and iron can all be bound to the phytic acid molecule, and then that decreases the bioavailability because once the phytic acid binds these minerals, it precipitates out and you're no longer able to absorb it, and it can't get into the unstirred water layer. Now, the majority of us, when we think of iron storage, we think that we think of the liver because iron is primarily stored in the liver. And but the intestinal mucosal cells, enterocytes, are actually store a significant amount of the iron, so they're able to absorb it. They have ferritin within them, so the iron is absorbed. It binds ferritin. And then if they don't need that iron, then when the intestinal cells are sloughed off, they just go into the intestine, and so the excess iron is then excreted with the rest of the feces. So liver stores iron, and the intestinal mucosal cells also store a lot of iron too. Now, our iron is, we hold on to it. Our bodies are really good at holding on to it, and we really try not to lose it. We try and have a closed system with our iron. So the main times in which we're going to be losing iron is when we have any sort of blood loss. So if you have cut yourself and you're bleeding, in addition, we have the sloughing off of the intestinal cells that are storing the iron for us if we have adequate amounts within our liver. But we are able to recycle the iron from the sloughed off cells. Those sloughed off cells, they're going to be digested just as if they were food, and so we're going to be able to reabsorb those later on in the intestine with the bile salts. Now, everybody is probably familiar with some sort of iron deficiency. So in humans, iron deficiency is the most common mineral deficiency that you're going to encounter. If you're deficient, usually it's due to some sort of chronic bleeding. So if you have some sort of ulcers, something that's going to cause internal bleeding, you're going to be more at risk for iron deficiency. Women are significantly more susceptible to iron deficiency, and that's mainly due to the menstrual losses. So you need to have, as a woman, make sure you take adequate iron supplements to keep yourself from getting anemic. And then you have infants and young children. They are not very efficient at converting the ferric iron to the ferrous iron, so that's just the development of the GI tract, so they are more susceptible to iron deficiency also. And then if you are undergoing rapid growth, you have a higher requirement for iron during those high growth periods. So those are some of the reasons why you're going to be more prone to iron deficiency. In animals, very rarely are we going to see iron deficiency, and that's especially those that are raised out in pasture. So our cattle, you definitely aren't going to see iron deficiency. We did see it in both the poultry industry and the swine industry when we went to conventional cages back in the 60s. So we went from our poultry and our pigs being raised out on fields and had access to the soil, and then when we put them inside on concrete floors, or with poultry when we put them in cages to reduce, and that was all done to reduce disease spread within the animals and keep them healthier, but we inadvertently saw some iron deficiency within them because they did not have access to the soil, and the soil has high levels of iron within it. So that was just a matter of incorporating more iron into the diets. Now, out of all the minerals that we're discussing in this class, iron is the most toxic out of all of them. And we see some toxicity. So there are some genetic components within it where we have hereditary hemochromatosis, and that affects 4 in every 1,000 people of European descent. Basically, those individuals, they have defective regulation of the iron absorption, so they are unable to control the amount of iron that their intestines are absorbing. So they really have to limit the amount that they have within their diet if you have that genetic mutation. And it ends up affecting the liver, the heart, pancreas, pituitary, and your joints. There's also Bantu, that's a type of hemochromatosis, and this is due to the overconsumption of iron-rich foods and water over extended periods of time. So this is basically a very slow overconsumption of iron over time. So it's more of a slow chronic toxicity that is occurring. So some soils are very rich in iron. So if you have your crops are mainly coming from there, your well water, you're going to have more iron that is brought up from that. And if it's not properly filtered to remove some of that iron, then you can have develop some iron toxicity. Now, the acute forms of iron toxicity is usually occurs more readily in children. So this is when they get their little paws on those fun gummy multivitamins that have adequate levels of iron, and they think those little gummy vitamins are are equivalent to candy. And once they figure out how caps work, and kids are very, very sneaky, you can think you have something well hidden, and they will find it and they will open it. Other times that you see it is when children end up getting into their vitamins and eat a whole thing of their multivitamins within a day, and it's going to be more acute iron toxicity. And then, since this is comparative animal nutrition, we do have our captive frugivores end up getting iron storage disease. So, if you ever see the toucans at zoos and other various frugivores, obligate frugivores, um, those animals, the fruit that they are eating in the wild has very low levels of iron within it. So they are constitutively always expressing the receptors to absorb all the iron and to transport it. So they don't have that negative feedback of the hepcidin saying our liver has adequate iron, no problem, shut it down. So basically, since they don't have that negative feedback, because naturally the diets that they adapted and evolved to consume had low levels of iron, now we have them in captivity and we are feeding them domestic fruit. And much of that domestic fruit has been bred to have higher levels of iron. And these animals are unable to downregulate that. So many of them end up dying of iron toxicity after about 3 or 4 years of captivity. Okay, next we're going to switch gears and talk about copper. So copper was discovered as a nutrient in the early 1900s. We had individuals that were anemic and they were treated with iron, and that did not reverse the anemia. So it was actually discovered at that point in time that copper is also required for adequate hemoglobin production and function. So some studies were done, and they showed that animals that were fed a milk-only diet, so only milk is what they had, and it was supplemented with iron, and these animals still got anemia. When they put copper in with the iron of the milk, then all signs of the anemia disappeared. So that's how it was discovered that copper is required to prevent anemia. So if you have adequate iron consumption, but you may inadvertently end up, but you're still anemic, you may have a copper deficiency. So why do we have copper? Why is it important? So it is a component of ceruloplasm, and this is a copper-containing protein, and it oxidizes ferrous iron, Fe2+, to ferric, and we need that the iron has to be in its ferric form to be transferred to transferrin. So we transport our iron as ferric iron, and it's bound to transferrin, but we store it as ferrous iron because that's less reactive. So if we do not have copper, we're unable to make ceruloplasm, and therefore we're unable to mobilize our stored iron from our liver because we're not able to then change it in between the ferrous and the ferric and transfer it to our tissues that need it. So copper is absorbed by active and passive transport in the GI tract. So if you are deficient to adequate levels of copper within your diet, you're going to be utilizing active transport. If you have excess copper within your diet, you're going to be absorbing it by passive transport. And one of the proteins that we need to absorb our copper is metallothionein. And we have the bioavailability of copper in general is pretty poor. It's not very well absorbed. However, the young individuals are much more efficient at absorbing copper than older individuals. And once we absorb our copper, we transport it primarily bound to ceruloplasm. So 90% is bound to ceruloplasm. Another 10% will bind albumin. And then when we have adequate levels of copper, we're going to excrete it via bile in the feces. So copper deficiency in humans is pretty rare. You're most notably going to see it in overall malnourished children. So if you have multiple deficiencies, that's where you're most likely to encounter copper deficiency. In animals, we have been able to induce it, and there are some places in which you can see it naturally occurring. And with that, we have they present with anemia, and that's because they're unable to interact with the iron to where it can properly function with hemoglobin. In addition, you have hair depigmentation, also called as achromasia. And this happens because copper is required to convert tyrosine to melanin. And melanin is the pigment that's going to give your hair or fibers their color. So we have hair depigmentation that occurs. In addition, in sheep, you could see steely wool syndrome. So the wool actually gets crimped, and that happens a lot of times if you have a interaction with molybdenum, which we'll talk about next. In poultry, you're going to have an aortic rupture occur, and this occurs because copper is involved in collagen synthesis, so you don't have proper cross-linking within collagen, and eventually that's going to lead to an aortic rupture. It's also associated with infertility, and that's because it is required for proper progesterone synthesis. So if we don't have copper, we're going to impair our progesterone steroid synthesis, and that results in infertility. Now, one of the main ways where you're going to see copper deficiency in humans is that there is a copper molybdenum interaction that occurs, and this is most notably observed within ruminants, our domesticated ruminants. So if you have soils that have high levels of molybdenum and even adequate levels of copper, but if they have the ratio of molybdenum to copper is high, then you're going to have your molybdenum actually antagonizes your copper, and regardless of the level of copper, you induce a copper deficiency. So basically what happens is you have sulfates react with molybdenum in the rumen. So the microbes are breaking down your sulfur amino acids. You have sulfates that form. Those sulfates react with molybdate, and it forms thiomolybdates. And these thiomolybdates end up reacting with copper, chelating it, and it makes copper unavailable to the animal. So then you get this change in hair color. So here you can see the bottom, we have the steer, and this is actually Black Angus. And you can see how that coat is more red, and that's due to the pigmentation. So we're now don't have the copper available, so we're unable to convert tyrosine to melanin because we need copper to act as a cofactor for that reaction. So then we have the change in the hair pigmentation. So if you work with ruminants, this is one of the more critical interactions that you will deal with on a more regular basis. And some soils have high levels of molybdenum, so you have to be aware of the amount of copper and molybdenum, and you may have to increase then the amount of copper within the diets if you have high soy molybdenum. There is a genetic disease in people referred to as Wilson's disease, and this actually ends up presenting in young adults. It obviously is a genetic disorder and slowly occurs over time, but once children hit puberty, that's when you start to see the ramifications of the disease and or when it presents, and it's associated with a defect in copper transport. So these individuals have low ceruloplasm. So they have low levels of ceruloplasm, so we're unable to transport our copper because, as you remember, 90% of our copper is transported with ceruloplasm. And they also have bile excretion ends up being impaired, and eventually, so you have low copper transport, you're not excreting the excess bile that you may have, and so eventually you're going to end up having accumulation of copper within the liver, brain, and cornea. So you could actually see in these individuals, their eyes are going to be more yellow. So it's not all individuals that have hazel eyes or have Wilson's disease, but you can see in this eye how yellow the pigmentation ends up around the eye. So you can see that in an individual with Wilson's disease. As far as toxicity, it's not common in humans at all. Usually, if you're going to see it, it's due to new copper pipes being put in, and with that, the symptoms include nausea, diarrhea. If it goes on for prolonged periods of time, you'll see liver damage. Animals have more reported cases of copper toxicity, and they're going to be more lethargic, have diarrhea, liver damage. And our domestic ruminants are actually more sensitive to copper toxicity than any of our other livestock or even wild ruminants.